This is Unit 3, Part 3. It's a continuation of neurophysiology. We ended up last time on this slide, and in the bottom left corner we have a diagram here of a neuron. The neuron has a central cell body as shown in the area here. And that's where the nucleus is, and then it has these extensions which are called dendrites, the small extensions shown around the cell body, which receive signals from other cells. And then the neuron would have it also has this long, and only part of it shown here, it can be extremely long, this long extended axon used to send signals out from the cell if it's activated. And that's where the impulses are sent. And we talked about in Unit 3A Part 1, we talked about the resting membrane potential, which if it receives a stimulus that's large enough, the cell can depolarize, and when it hits threshold, it will trigger an action potential sent down the axon. So that's how impulses are sent out by neurons to other neurons or to excitable tissue like muscle tissue. So if it hits the threshold, then you trigger an action potential, in which case sodium channels open. You get depolarization, in other words, movement up towards the more positive. But that doesn't just keep going more and more positive. With the sodium channels open and sodium rushing in, they then close, and then potassium channels open. Because potassium is higher inside than outside, potassium starts to leave, and you get repolarization. Going towards more negative, you get a bit of a hyperpolarization, and then back to the resting membrane potential. And that return to resting membrane potential is established by the sodium-potassium pump, which is constantly pumping sodium out and potassium in. Remember, those pumps are not the same as channels. So here you see on the next slide the changes that you get in the channels being opened and closed, and permeability indicated by P of sodium, and that's the yellow or orange line, the permeability of potassium, which is the green line, and the actual level of membrane potential, which is shown in the blue line. So for the blue line here that shows membrane potential, you use this scale on the left-hand vertical axis, and for the different permeabilities you use this scale, you see, on the right hand. Because after all, permeability and membrane potential are completely different things, different units. So as I said again, you have a resting membrane potential, and that's shown right there. And it's around minus 70 millivolts. There's some variation depending on the source you see. You can get depolarization with a stimulus. If a stimulus comes in, then you get depolarization. The stimulus could be all kinds of things. Within the central nervous system, it's typically between neurons. But if we're talking about a sensory neuron that's sensitive to light, could be a light signal that causes depolarization. If it's a baroreceptor in your aorta, in the aortic body, right? Baroreceptors that are sensitive to pressure, then it could be just the stretching, the pressure triggers the action, is the stimulus. So with stimulus, you get, if it's big enough, and then you get depolarization, and when you hit the threshold, the sodium gates open, and you can see that the sodium permeability, it rises at that point, shown with the orange line. So the sodium permeability is rising. That's because channels have opened. Sodium gate opened, so the channel opened, and so sodium is rushing in, making the inside of the cell more positive. You get an action potential. Then they close, those sodium channels close because the inactivation gate closes, and you get repolarization because what then happens is that potassium gates are opening. You can see the green line that the permeability of potassium is rising right there right at this point here in time. So potassium channels open, potassium starts to rush out, the membrane potential starts to go more negative. You're getting a repolarization, and the action potential is now on the downward curve. I'll draw it here. You see there's the action potential, resting membrane potential, depolarization to threshold. Then sodium gates open, sodium rushes out, so it's depolarizing. Then they close, and potassium gates open, potassium is rushing in, it's positive, so it becomes, I'm sorry, I got that wrong. Potassium is rushing outwards, so the inside becomes more negative. And then it goes below, that's a hyperpolarization, then that gets corrected with sodium potassium pump activity and goes back to the resting membrane potential. Okay, so remember, on the rising phase, sodium is rushing in, and then on the depolarization and with the repolarization, potassium is rushing out, so you get more negative on the inside. And that action potential is triggered, and I'll explain to you later, it actually travels very, very rapidly down the axon, right to the ends of the axon. Okay, let's talk about that mechanism of sending the action potential down the axon. Propagation of an action potential. This is zero time. All those depolarizations and repolarizations and movement of electrolytes that I was talking about, or ions, sodium, potassium, are all occurring at the level of the membrane, just inside, just below the surface of the membrane, right at the base of the axon, where the axon just first emerges from the cell body. So if this was a neuron, in other words, a nerve cell with some dendrite extensions, and if the axon is here and goes like that, way out there, then the stimulus comes in and the key is at the base of the axon, which is called the axon hillock. If it has enough of a stimulus, then you get enough of a depolarization. What you get is the sodium channels open, and sodium starts to move in. Okay, and that's all happening right just at the membrane. Okay, it's just coming into the membrane, just coming in at the base of the axon hillock into these areas. The sodium is moving in, so the inside becomes more positive. Sodium gates are opening, and you'll see in the next slide. So this is the distance along the horizontal axis here. It's the distance along the axon, starting with at this end down here, right at the base of the axon where it joins the cell body, so the axon hillock, and then this is further down the axon. And this is the membrane potential that you see at any one position. Now, if the neuron was stimulated enough to trigger an action potential at the base of the axon, where it joins the cell body, then those channels opened, and now they're just closing, and the action potential starts to move, or it's called to propagate down the axon. So the next section of the axon gets channels opening, sodium channels opening, and sodium rushing in just below the membrane. Here you have the membrane right in that, pictured right in this area here. So at the right across the top there, in this section here, what they're showing is what the membrane looks like at different distances along the axon. So this area was open and now they're closing, and now this area opened, and further down the axon, these channels, these sodium channels, gated sodium channels, are still closed. And if you go, this is right at the time of the initial, it's just a tiny bit past 0 milliseconds, because this area here did get stimulated. Okay, so 1 millisecond later, after the previous slide, after some sodium ions have moved into just inside the cell, just underneath the membrane, they get attracted and move to negative charged areas that are further down the axon, and that's a current. And that current depolarizes that area just further down the axon, so it depolarizes to get to threshold, and action potentials are generated in that region, and sodium channels open in that area. So now we're further down the axon, along the axon, than the last slide that I showed where the action potential was in this area. But now those gates are closed, and let's go back to resting membrane potential, and now the action potential is shown here as being further downfield down the axon. Sodium that moved in through the previously open channels in this area have now moved into here and caused sodium channels in this area to open. And so in this way, the action potential moves away from the stimulus that was experienced at the base of the axon hillock where it joins the cell body, and moves down the axon. I should point out where the sodium channels were just closing as the action potential passed through a particular area. Potassium gates are opening, and that's what's creating the repolarization, so that the inside of the cell just under the membrane is negative. Okay, so in this area that has gone back to resting membrane potential is because of an inrush, a rushing out of potassium to bring it to more negative. And in this way, it moves down the axon with time. I should point out the threshold is about 15 to 20 millivolts above the normal resting membrane potential. So in a graph with membrane potential down on the vertical axis and time along the horizontal axis, if you look at the membrane potential, if this is the threshold, right, along here, say around minus 55 roughly, and if this is the resting membrane potential, it's going to be about 15 to 20 millivolts below the threshold. You get depolarization, and is up to about 15 to 20 millivolts higher, and that will trigger the action potential that we just talked about. Okay? So basically, there is a flow of charged electrolytes or ions. That's the current. It's flowing first into just underneath the membrane, and then along just underneath the membrane inside the cell. If a stimulus comes in that's below threshold, then action potentials are not generated, and you don't get that propagation of the action potential down the axon. If strong stimuli come in and hit threshold, they trigger an action potential, and the action potential propagates very, very rapidly down the axon, and it's always an all or none thing. It will happen or it will not happen. It won't vary in size. It'll always be the same size. So they're all alike and independent of the stimulus intensity. What you need is a threshold stimulus. Once you hit threshold, you'll get the action potential. All those action potentials will trigger, will be the same and propagate or travel down the axon. The central nervous system will be able to respond to the intensity of a stimulus by the fact that there'll be an increased frequency of action potentials or impulses. So they're coming more often with a stronger, if you have say a weaker stimulus onto the cell, then an action potential is sent down, and it has, a certain activity or impulse strength. If you have a stronger impulse coming in, you'll still get the same kind of action potential, but there'll be more of them, one after another, going down. The frequency will be faster. So the central nervous system can determine strength of a stimulus, not so much by the size of not at all by the size of an action potential that's generated in a neuron, but how often the action potentials are generated. So here you see the upward arrows in red that are shown here. For example, these are each arrow in red is a stimulus, and then the downward arrows when the stimulus stops. Here is a subthreshold stimulus that was unable to reach the threshold depolarization, so no action potential was generated. Here a bigger stimulus, and it's a bigger stimulus because the arrow is longer, did hit threshold, and an action potential was generated, and with that stimulus then another one, and they travel down the so if you're measuring to see whether action potentials are being sent out down the axon, yes they are, and the frequency is shown here. Every few milliseconds time, less than a millisecond time, you're getting action potentials generated, but they're all the same size. They all hit the same level of depolarization. Action potentials are always the same. With a stronger, and here of course it ends in this area here, the stimulus stopped, but then the next stimulus was bigger, so you get a faster, higher frequency. Frequency is per time. Action potentials are the same, but they're closer together in time. Frequency goes up, same, and then turns off. Back to resting membrane potential. Bigger stimulus, it goes not higher. The peak here is not higher. Don't worry about this area down here that's going up. That's irrelevant. It's the that don't get confused by that. It's how high this action potential depolarizes, and it's always the same. The only thing that changes is how often an action potential is being sent down the axon. Okay, so action potentials are always the same, but with a stronger stimulus you get a higher frequency of action potentials. And this is exactly what I said before, that the strength is shown by the length of the arrow. There is something that you should be aware of, and that's called an absolute refractory period. There is a period of time after the opening of sodium activation gates until the closing of the inactivation gates, during which you cannot trigger an action potential, and that's the absolute refractory period. So that you cannot generate another action potential during that period of time. That mechanism guarantees that each action potential is a separate impulse coming down the axon, and it helps to actually allow for transmission of the nerve impulses going down the axon of a neuron. If this is a neuron, right, cell body, then it guarantees that there's only transmission of action potential in one direction, down the axon to the end of the axon. It cannot, this absolute refractory period prevents travel of action potentials upstream, or it's called retrograde transmission. It does not happen, ever. And here again, the same, just to remind you, the same, diagram showing a action potential being generated with a stimulus, and the different channels open, as you can see by the sodium leaving, and then sodium entering and potassium leaving here during repolarization. And at the top, you can see that there is an absolute refractory period of time during which you cannot get another stimulus. And then that's followed by a relative refractory period. So on this point here, once it hits, I didn't draw it very straight, but right here, once it hits that threshold and is below the threshold, you can get, even though it hasn't completely recovered back to the normal resting membrane potential, you can get another action potential generated during this time, although the stimulus would have to be a stronger stimulus in order to trigger that second action potential. Okay. Here, of course, after the end of the relative refractory period, when it's back to the resting membrane potential, it's the same, exactly the same as anywhere in here where a stimulus can depolarize and hit threshold. Okay, during that relative refractory period, what is in fact happening is that the sodium gates are closed, the potassium gates are open, and repolarization is occurring. Remember, what's happening, in fact, is that we're talking about the period of time when it's in fact becoming hyperpolarized and then moving back towards that resting membrane potential time. Okay, so this area in here, that time period is the relative refractory period. Okay. Sodium gates are closed, potassium gates are open, and repolarization is occurring, going down. And in fact, it has to go through that return back to the threshold. At this point, the threshold in here, in this time period here that I showed, okay, during that time period, the threshold is further away. If threshold is here, okay, then normally from resting membrane potential to threshold is 15 to 20 millivolts, but in this period of time, it's further away, so only a stronger stimulus can increase. Only a much stronger stimulus is needed during the relative refractory period to reach threshold. So what you have is a relative refractory period where a stronger impulse could depolarize up to threshold, but it takes more to get up to threshold. Threshold is further away, is larger. The distance to threshold, as shown here, right, is bigger, requires more of a depolarization than usual. So that results in a relative refractory period. Okay, movement of the action potential down the axon, the velocity of it can vary. And it varies actually quite widely between different types of nerve cells or neurons. The velocity, the rate of movement of the axon down, of the action potential down the axon, is the impulse, the rate of impulse propagation or conduction of that impulse down the axon, and it varies depending upon the diameter of the axon. The larger the diameter, the faster an impulse moves. The other key determining factor is what's called a myelin sheath. Myelin is a lipid-based molecule that dramatically, the sheath, I'll show you the diagram again that shows the structure of the myelin sheath around the axon. Myelin, presence of a myelin sheath, also called myelation of the axons. So myelation will dramatically increase the speed or conduction velocity, speed of the impulse going down the axon. What happens, in fact, is that the current passes through a myelinated axon faster because it only has to move or occur at what are called the nodes of Ranvier. The nodes of Ranvier are the places where the axon is not myelinated. So in between the myelin sheath, there's a series of myelinated areas along the axon, and the nodes of Ranvier are where there is no myelin, and the signal or depolarization essentially jumps along just underneath the membrane. It jumps from one area to the next, from one node of Ranvier to the next. You have, as you know, voltage-gated sodium channels. They're concentrated at the nodes of Ranvier. And so action potentials actually are not triggered all the way down a myelinated axon. They're triggered sequentially down the axon only at the node. So the action potential essentially that's initiated at the base of the axon, right at the axon hillock where the axon joins the cell body of the neuron, it will occur in a jumping kind of manner from one node to the next. That occurs much, much faster than along an unmyelinated axon where you have to get all the events occurring in sequence all the way along the surface of the membrane, all the way down the axon. In this case, you only have to get this area here at the base has to reach threshold. That opens up the sodium gates, as you know, and sodium rushes in, and so you get depolarization, and threshold is met, and action potential is generated, and that spreads inside the membrane to the next area, which is called a node of Ranvier. These are the myelin sheaths. I'll color it in. There's a myelin sheath, there's a myelin sheath, all this, and then this, and then this, and this. So it's myelinated all along by the sheath. It's actually it occurs during development of the nervous system. They get myelinated, and it's in this kind of sequential regions along the axon that are myelinated, and the action potential does not have to get generated everywhere. It all along the membrane. It only has to occur at the node of Ranvier. there's enough spread, as you can see shown in this area here of the diagram. There's enough sodium that moves in when the sodium gates open. Okay, if the action potential is reached this point, remember the sodium gates open, sodium moves in. Enough sodium moves into this area just under the membrane to spread along to the next node of Ranvier, triggering triggering an action potential at that point. And again, sodium channels opening with that action potential. So it's essentially, if you look at it, the action potential is occurring here, and then there, and then there, and then there. It's essentially jumping along from one section of the next down the axon. And this kind of conduction is much faster than if you didn't have if the myelin was not present. And this kind of conduction of the action potential down the axon is called saltatory conduction. Saltatory means jumping. So saltatory conduction is much faster. If the diameter of the axon is larger, it's also faster. Those are the two key things, diameter and myelination. Now there is a disease that affects young adults. It's quite a terrible disease that affects young adults. It's an autoimmune disease where the immune system attacks in a person. Normally the immune system only responds to foreign non-self molecules. In this case, it's an autoimmune disease where the immune system actually attacks and destroys the myelin sheath of the nerves, and you get a serious kind of disease which is called multiple sclerosis. It's essentially a short-circuiting of nerve impulses. Okay, now the last slide is, this is one disease of many that can occur that are disturbances of the nervous system, but it's specific to propagation or conduction of nerve impulses, and that's why I'm discussing it. I bring it up. Very minimal description of the disease. There are disease-modifying drugs that can treat symptoms, although there is no cure for the disease, but it can reduce the complications, reduce the disability associated with them. And not all, but some live for quite a long time with some disability, but they are making progress in understanding the mechanism of the disease, and with understanding the mechanism, there's more probability for finding a decent treatment, or at best even a cure, but they are working at it, and there are some drugs that can modify this kind of destruction of the myelin. And that's the end of Unit 3, Part 3.